My name is Simon Vukelj, and I am the Director of Communications for the Children's Tumor Foundation. This evening, I am also your moderator. Tonight, there are over 300 people listening as we at the Children's Tumor Foundation take you through Foundation priorities for 2013 and beyond. Our featured speakers this evening are the Foundation's President, John Risner, and the Foundation's Chief Scientific Officer, Annette Bakker. John Risner rejoined the Foundation as President in 2012 after having previously served as President from 2005 to 2010. Prior to that, he was a member of the Foundation's Board of Directors, and he also serves on the Congressionally Directed Medical Research Program, NF Research Program Integration Panel. Annette Bakker joined the Foundation in November 2011. Dr. Bakker has 25 years of experience in research, including 10 years in basic cell biology and 15 years in drug discovery and clinical development. She received her PhD in biomedical sciences from the University of Antwerp in Belgium and was a fellow at Yale Medical School. Dr. Bakker has also worked as a group leader for oncology at Johnson & Johnson and as a director of R&D at Siena Biotech in Italy. Before we get started, I'd like to share a few housekeeping items. After John and Annette have given their presentations, there will be a question and answer period. Because there are several hundred participants on this call tonight, all phones have been placed on mute. You can submit your questions in the text box to the right, and we will answer as many of them as time permits. Most of you are likely listening to this broadcast over your computer speakers. If you are having trouble with the audio and have already adjusted the settings on your computer, you may want to use the call-in phone number up on the screen. It was also given in the confirmation email you received when you registered for this webinar. Next, this event is being live captioned, and the text can be viewed at ctf.org/caption. Please note that you will need to open a second browser window, one in which to view the presentation and the other to read the captioned text. Tonight's webinar is being recorded and will be available on the ctf.org website this week. And with that, let's get started. Our first speaker is John Risner, President of the Children's Tumor Foundation.
Great. Thank you, Simon. The next slide. I think a lot of people on this call, most everybody knows about the Foundation, so I really want to focus this on what the research priorities are for 2013. But I will share that our overall aim is to serve our one and only client, the patients. All of our efforts are aimed at getting the best treatments as fast as possible to the patients. Every day that we come in, this is what we think about. Some days that focus maybe is in advocacy, other days fundraising, some days clinical issues. But nonetheless, there's a variety of efforts that are made here, but they all have one goal. And really, and shortly, to put ourselves out of business. By serving our patients and truly ending neurofibromatosis through research, I'm confident we'll be successful. Who we are and what we are not. Well, who we are is we are a bridge between all those who want to offer a better life to our patients. We are strong collaborators. Who we are not competitors. Now, competition is a great thing, and we feel the competitive edge every day, but not in our research program. We know that we will not cure NF on our own, not NF1, not NF2, nor schwannomatosis. To really achieve our ultimate goal, we need to fiercely collaborate with industry, with academic labs, with other organizations. At the bottom of your slide is just a logo from Faster Cures. This is an organization based in Washington, D.C. that really took an interest in helping all research-based foundations collaborate among themselves and set some templates. For example, we negotiate regularly with academic labs and industry in terms of patent policy and intellectual property agreements. By helping create a clearinghouse, templates that can be used, commonality among different organizations, they've really helped cut through a lot of the red tape and are very much thought leaders in the nonprofit community, and we're very proud to be part of that organization. Our strategic direction: invest in critical efforts that accelerate the translation from research findings to clinical benefit. Now, while this might seem simple, this is really a change over the last 5 to 10 years. Certainly 10 years ago, and as recently as 5 years ago, there was very little known about the underlying biology of NF. And if you think in broad strokes of what the Foundation has done, after being formed in 1978, really the research got rolling from 85 to 95, and a lot of that research was spent just really on just basic biology. Once the genes were discovered for NF1 and NF2 in the early 90s, and certainly with the advent of the Congressionally Directed Medical Research Program and adding NF in 1996, the focus there turned to translational research, to developing animal models and cell lines to really learn more about NF. And since the 2005-2006, with the establishment of the Clinical Trials Consortium, again by the CDMRP, there's been a real push into clinical research. While not entirely accurate, the clinicaltrials.gov site, if you search that site for NF trials back in 2005, I believe there are 4 that will show up. If you search that today, you'll see more than 40. Now, some of these are natural history studies and not truly, therapeutics that are into the clinic, but still there are over 20 compounds that are being tested across a variety of NF complications today. We see our job as really being fostering that, accelerating that, and do everything we can to speed those treatments to the clinic. Our mindset begins, as all research does, it begins and ends with the patient. You see a kind of diagram here, and Annette will walk you through some of these specific steps and what we're doing in these areas throughout the presentation. But really, it starts with the patient. It goes through tissue, basic science, understanding what you're working on, developing compounds to treat the complication, using preclinical test systems before it ever goes into a human, and coming back to the patient. One of the key things about this is not so much a compound itself, but is where are the patients? While we have a large database here, it is not exactly set up to record a lot of data on the NF complications each patient faces. So beginning in 2012, we launched the NF registry. Now, this is hosted, actually contracted out by Patient Crossroads, so it's not held here at the foundation. They are an organization that does this for several other disease groups and has a lot of experience in this. The primary aim of this is to connect the patients with appropriate clinical trials. The secondary aim, and really this is probably just a key finding of this, is that pharmaceutical and biotech companies, in order to get interested in investing in NF research, need to be assured that if they have a drug that they want to test, let's say for plexiform tumors, that they can, in a short order, fill up a clinical trial. The trial recruitment phase, a short one, takes about a year to a year and a half. But a pharmaceutical company, if they find they cannot fill their trial, or a trial that they budgeted to take a year to recruit for ends up taking 3 years, that's not a disease that they will probably likely continue to work on. So getting access to the patients is a key variable. And some of the other leaders that we benchmark ourselves against, certainly Cystic Fibrosis Foundation and Multiple Myeloma Foundation being 2 of the prominent ones, have really shown the value of developing these registries. The data is highly protected. We do not have access to it here at the foundation. But this was launched in late 2012. We are growing. Today we have 1,000 registrants. Anybody on the call here tonight who's already registered, thank you. Anybody on the call who has not registered, please visit the site. And I'm going to ask Simon, if you would, click on the hyperlink there, and our screen should change. You can see, I'll just show you the home page of this right now, just briefly, because this is such a key tool for research. So you can see it's not hosted here, it's hosted at Patient Crossroads. This registry is for patients of NF. Now, for adults, we encourage them to register directly. For children, we encourage parents to register for them. Your email address will be recorded. And what will happen with this information is if there is a clinical trial that appears to be of benefit to someone with a complication, the email will go back to them. It will not go to a doctor. It will certainly not go to a pharmaceutical company. It will go back to the patient. And the patient is then encouraged to ask their doctor, oh, there's a new trial that I saw that's opened up for Gleevec for plexiform tumors. I have a plexiform. Would this be of benefit to me? So it really is there to help the clinical community and the patients to generate dialogue. As we go back to looking at what we have patient-wise in the United States, it's estimated that 120,000 people have NF1. I don't think while it's a large number, we expect to launch a full campaign to drive registrations in May, and our goal for the end of the year is 10,000. Now that is less than 10% penetration of the NF community, so it's a matter of just us getting the word out there. So look for this campaign, and again, I encourage you strongly to please visit the NF Registry website and register if you have not already. Fostering and working on the registry and also working with the clinic network, I'm very happy to report that after a long search, Pamela Knight, a former genetic counselor, joined us in January. Pam will be responsible for working with the clinic coordinators at the 44 NF clinics in the network across the country, but also fielding a lot of questions from individuals, from patients, and even from doctors in rural areas that might not have the level of expertise of NF that you find in some of the major centers. Her focus will be to connect between the NF Forum, patient symposia. She informs the bridge between the science and the patients. Pam, before going into genetics counseling, was actually a writer for the science magazine Nature, so she has a great deal of experience in translating complex scientific information into lay language. And of course, she'll be available as part of our support team along with our volunteer relations staff that can help with any patient questions that come up. With that, I'd like to turn this over to Dr. Annette Bakker. She'll get into some more of the deeper research areas and walk you through, and there'll certainly be time for questions and answers at the end.
Thank you very much, John. So as John pointed out before, everything starts and ends with the patients. So the first part is knowing where the patients are and being able to identify those patients for the drug discovery process to start to take place. The first step before we can even start identifying potential treatments for patients is, of course, to understand the biology of the tumors and all the other potential problems our NF patients have. And therefore, we need basic biology. But in order to do the basic biology, we need, of course, the tissue. So although there are quite some biobanks out there, CTF is now launching a specific biobank. I will go back. Yeah, can we go to the next slide? So, and the aim of that biobank is to be able to offer an opportunity for everybody to have the tissue stored in a place where it is safe. And although it may sound simple, but the tissue, in order to be able to be useful, needs to be collected in a very certain way, in a very specific way, always the same, and those are called standard operating procedures. And then the second thing of this tissue is, before being really able to analyze what is inside, you need to be able to store that under extremely well-controlled conditions. And there are very specific service providers that do this, that offer this kind of service. So to come to our specific biobank, our biobank is a global biobank, and the idea is that our biobank would be project directed. That means that we would collect the right tissue in the right format so that it can be used immediately after that. And we would first identify what kind of project we would be interested in, and then collect the exact right tissue. We are now launching our first pilot project, and our first pilot project is on dermal neurofibromas. They will be stored at such a specific company that is able to make sure that the necessary quality control is guaranteed. The company is called Precision Bioservices. We are collaborating with a plastic surgeon who is specialized in dermal neurofibromas. And as I told you, our biobank is a project-based biobank. So the project of what we will be doing once the tissue is collected is already on the table and is already decided what we're going to do with that tissue. So we are really making sure that we isolate and that we obtain and collect the exact right material that we need in order to perform this study.
Annette, if I could just add something here. One of the importance of this is, I don't know if everybody read about it last fall, but Harvard University lost power to one of their freezers, and there were 10 years of samples that were thawed out and destroyed as autism samples. And then again with Hurricane Sandy here in New York, the water flooded the NYU basement, which held their tissue bank, and also cut off the generators that fed their mouse colonies basically. So looking at this has been a long project to look at this and find a site that is as protected as it can be, but also has redundant systems that hopefully, as you build this bank, you have something that you can have is 99.9% assurance that that your tissue samples are safe, and you won't face a situation where 10, 20 years of data are lost to some cataclysm.
Yeah, exactly. Thank you, John. So once we have the tissue, in fact, our role is to fuel the science. We are fueling the science by funding grants, awards, and also collaborating using contracts. And for most of you, probably these three grants or awards are familiar. They are the Young Investigator Awards, abbreviated as YIA. Those are typical basic biology science grants. They're very innovative. And I mentioned the word seed funding, and that is in fact the reason why I put that on the slide is that very often once people got the initial funding from our foundation, they go to bigger organizations such as the NIH, such as the Department of Defense, to really get access to bigger funding to make the major discoveries. But the first seed funding is essential in order to get this data that you need in order to get granted from the bigger organizations. The next initiative is the Drug Discovery Initiative, and that is, as you can imagine, close to my heart. That is, in fact, those awards are meant to fund translational research. And I suppose many people have heard about the word translational research. And in fact, translational research means the research that you need in order to translate, between brackets, the basic biology finding into something which has some clinical benefit, which can then be brought to the patient. So it is really an essential part of biology that is needed in order to get treatments to our patients. The last one are the Clinical Research Awards. Those are all kind of awards that are associated to clinical studies. So they can or be short pilot trials, they can be clinical enabling studies, so everything that is needed to make a clinical trial more informative and better. They are, again, innovative, and in the same as the Young Investigator Awards, the idea is really to be, again, a seed funder so that people can get access to bigger funding if necessary.
And I would just add, Annette and I were at a conference held by Faster Cures last fall. So this distinction between basic science, which is biology, and clinical science, which is drugs that go in and treatments to patients, it's the translational piece in the middle. And there was an industry panel, and one of the representatives from a biotech firm, he defined translational science as research that does not result in a paper or a profit. And that was just such an elegant answer, because the publication of papers, that is how academic labs are measured. That is how science advances. So that is what they are very keyed into, is what projects they're working on will result in a paper that will make it into Science or Nature, or one of the top publications. Clinical science is the province really of biotech and pharma. They want to invest in things that will have a very quick financial return, get into the market, have a financial return. It's the translational area in the middle that is really sorely in need of funding, and that's where we really focus our work. So while we're covering all the three areas here, really in terms of dollars, the bulk of our dollars are going into the drug discovery area in the middle, into that translational realm.
Exactly. Oh, I'm sorry, I have done something that I shouldn't. Okay, I'm sorry, I was clicking on something where I'm not supposed to click. Okay, so if you would like to know which awards we did fund and where did we spend your money, the money that most of you people, and for which we are, of course, extremely grateful, have been raising for the foundation, I have worked together with an organization called Thomson Reuters. And Thomson Reuters is everybody knows Reuters as the journalism company. But Thomson Reuters is, in fact, a company that is specialized in pharma and biology information. And we have been building together with this company disease maps. And a disease map is, in fact, a diagram of the disease. And on top of those diagrams, there are little dollar signs. And if you click on the dollar signs, you can, in fact, see where CTF has been funding. So, yeah, Simon, thanks for opening the hyperlink. So if you go down to disease maps, basically you see it has been subdivided into NF1, NF2, and schwannomatosis. And if you look in NF1, it has been, just for practical reasons, to be able to put it on the screen. For NF1, it was subdivided in NF1 in tumors, NF1 in nervous and vascular system, NF1 in bone, and then there is the last one, which is called the dashboard, and I will show you in a minute what that is. So you see here on the left part of the screen, you see a kind of diagram of the disease. It's a high-level diagram, not very detailed, and of course there are many processes that we still don't understand. But if you look here and you see here, total 37,000, total 441,000. So you see these little dollar signs. If you click on those, you can see on the right side of the screen, you can basically understand. And so the 441 that you see when you click on the mTOR signaling, you can basically see all the grants that we have been funding by the Children's Tumor Foundation. So that is one way to look at it. And you can do that for tumors, you can look on nervous system, you can look on bone. You can do that for NF2. So it's the same story. If you look, for example, here, schwannoma, $653,000. Here you can see where this money has been spent. Schwannomatosis, although there is still a lot of biology to be done, and as you can see, there is a lot in the early phase of the disease, in the gene description, and then of course there is a big investment in Schwann cell biology. But there is still, as you can see, there is still a pretty open diagram here where not many dollars have been invested in these systems. So in fact, these schemes will not only help you to understand where we have been funding, but it will also help people understand where we may need to be funding for the future in order to make progress in the disease. And then the last one, which is, I think, a pretty important one as well, is in fact what I would call a drug discovery, drug development dashboard, where you can see, like, okay, how much have we been spending on cell model development in order to find better treatments? We have been spending huge amounts on mice. We have been spending quite some money on, well, not a lot, little money on screening. We have been spending quite some money on preclinical, and the reason why we have in these areas relatively little is because we had the NF preclinical consortium. I will explain you in a minute. And we have been investing in biomarker studies, in population studies, in early studies in humans. So you can see that we have across the platform, both methodology-wise as well as disease-wise, you can really see where we have been funding, and I think it's pretty nice to play with. You can find this on our website, ctf.org, about the foundation, and it is updated every time we have a round of grants. Let's say maximum a month later, the grants are in the disease maps.
And I would just add, Annette, that this tool was developed really for the scientific community, because when we talk about collaboration, what we want to do is eliminate duplication. And we want to make it easy for the scientific community that may be working on an allied disorder. Maybe it's a form of cancer, maybe it's a sarcoma. For them to see very quickly on our website what's been done already, who are the key thought leaders in it, and who are the potential collaborators. And so as we developed this, we originally had it behind a password system for the scientific community, and we realized, this is the type of thing that we should open up to the donor community, so you can see exactly, just like Annette said, thank you all for the support that has made this possible. You can see where this is going, and if you have questions about this, shoot them into Simon, and he'll get them answered appropriately.
Yeah, and then that's true, John, and there is one additive thing that is really important, is that for the moment I'm trying to convince industry partners to be interested in Annette as well. And as they see pathways or disease diagrams that they're used to see, and they may have molecules in those disease pathways, it might be more interesting to get the companies interested in neurofibromatosis. Okay. So, as I said, the Children's Tumor Foundation is fueling science by funding and by fostering. Just a quick example on fostering genomics work. So malignant peripheral nerve sheath tumor is a very rare condition of NF1, and it is in fact a malignant transformation of existing tumors. And we know something, but relatively little about these tumors. And we have been very actively lobbying to include this MPNST tumor analysis by the Cancer Genome Atlas program. And Kenna Shaw has been of extreme help here in making this happen. And I can proudly announce that today they signed off to do the full analysis of these 50 malignant peripheral nerve sheath tumors. So basically, they will throw the book at these tumors as part of a very big sarcoma project. So not only will we learn about these malignant peripheral nerve sheath tumors, but will we also maybe see connections between these tumors and other sarcomas. So maybe, John, you want to add
I just want to say that this was, Annette joined the foundation in late 2011, and she had heard of this project in early 2012 going on for sarcoma. And again, this is probably going to be $10 million spent by the National Cancer Institute. And so she called him up and she said, look, I'm working in neurofibromatosis. We have a form of cancer that is a highly morbid form, and we want to be included. And at first they were kind of reticent, but she kept hounding them, kept working with Kenna in a nice way. And then it became when she said, okay, we will do this if you can guarantee me 50 samples by the end of the year. Now, 50 samples with and the samples needed matched blood as well. And unfortunately, MPNSTs, they are one of the more highly mortal conditions of NEF. While fortunately it's rare, a lot of times the patients pass away without blood being stored. So securing those 50 samples, getting them sent to NCI, meeting the deadline. I want to take this opportunity, I'm sure Annette would too, to thank all the clinicians out there that were able to share their samples. So this is an example of work that they're going to spend anywhere from $500,000 to $1 million on analyzing these samples. That is money that we don't have to spend. We can focus on other things, but the value that will come out of this project is going to be just a tremendous for the NF1. So.
Exactly. And then in addition to this, the data will be completely open source, so will be completely openly available to everybody, which I think is also a major breakthrough for neurofibromatosis. So this is another initiative that I started this year in the Children's Tumor Foundation, and I think there was a ton of potential scientific opportunities in which CTF would play a more active role rather than a funding role, being really a partner in the science. And as I am still a very motivated scientist, I felt very motivated to get something where I can still use my science part of my life to advance neurofibromatosis. So, in fact, the idea is, together with our patent attorney, Marco Nieto, we developed a model in which CTF can initiate or can collaborate with academic labs, just like academic labs do between each other. We do that in a collaborative manner, and we work together with a company called Phoenix, who then subcontracts to the Children's Tumor Foundation and in fact executes very highly technical experiments in order to give us the information that we need to kind of drive the collaboration. So Phoenix is a company that is specialized in what they call high-throughput siRNA. So it's in fact a technology that is highly interesting if you want to identify novel molecular mechanisms for a disease. And together with Chris, we are now collaborating with, and as you can see, with 2 academic labs in order to make sure that this happens. And these collaborations are both in NF1, so there is one project that is specific to NF1, and there is one project that is specific to NF2. So I think we will be able to announce some very nice results very soon. We are very hopeful. Things are going very well, and this is only a start, these 2 projects, and the scientific community will really be invited to become part of this exciting project. Exactly. Sorry. Okay. Next. So if we go now to the core of drug discovery, there you typically need compounds, and you typically need test systems in order to make sure that your molecules do something to the cell, do something to the mice before you can bring them to the clinic. So the things that are ongoing now is a compound scouting initiative. So we have identified molecular mechanisms that would be important for neurofibromatosis. We are identifying compound opportunities with different companies. We are connecting compound providers and academics. And for the moment, there is already some proof of concept testing ongoing under a contract with the Children's Tumor Foundation. So in order to be able to test the molecules in the appropriate models, although there is a lot of discussion about what the value of a model is, and as John rightly pointed out before to me, we're not here to cure mice, but on the other hand, the mice can be extremely important models for us to understand better the disease and to test a compound in what I call a whole organism model. We need models desperately. So we need to develop the cell and animal models, and then we need to fund the testing of the compounds in the animals. So therefore, a couple of years ago, in the Children's Tumor Foundation, the NF Preclinical Consortium was set up, which is a unique consortium of academic labs that work as a team. And I emphasize the word team because I think it is really important as a move towards collaborative initiatives. And these academic labs have all of them have a relevant animal model that is relevant to neurofibromatosis. And they're testing what they call repurposed drugs. So repurposed drugs are in fact drugs that today are under development for other indications. And they are now tested for NF. Once, of course, it makes sense. They are rationalized for NF, then they test them. And once the compounds are what they call de-risked, the idea would be to go with these compounds into the clinic. It is the largest R&D investment of the foundation. As you can see, it's around $8 million. And this project is completed in June 2013. That means that since the results are nice, we have multiple clinical trials that were driven by the efforts of this preclinical consortium. We are absolutely convinced that the work should go on. So we have a collaboration agreement with the Neurofibromatosis Therapeutic Acceleration Program at Johns Hopkins, which is led by J. Blakely. And for NF1, we will collaborate with NTAP. So the animal models that are part of the NF1 consortium are plexiform, dermal neurofibroma, malignant peripheral nerve sheath tumor, and GMML. And for the NF2 consortium, that will be entirely funded by the Children's Tumor Foundation. And there the idea is to set up a highly collaborative effort from the biology all the way to the clinic. So as you recognize this scheme from the earlier slides, but in fact all the efforts go back to the patient. So once the compounds are good enough, safe enough, active enough, then they can be tested in patients. So the Children's Tumor Foundation has set up a NF clinic network, which are 44 clinics across the United States. We are fostering integration of these NF clinics, and we have a meeting for genetic counselors, clinical care providers, nurse practitioners, and that is going to happen in Nashville very soon. We're funding clinical research, and we are strong supporters of the RAINS initiative. And RAINS is, I think, a very important initiative. It stands for Response Evaluation in Neurofibromatosis and Schwannomatosis. And the idea there is that clinicians would sit together to agree on the way they diagnose NF and on the way, and schwannomatosis, and on the way they measure success of molecules. And that, I think, is an extremely important step towards clinical development of novel molecules. As I said before, for the integration, we would really like to invite the people that are sitting on the other side of the screen and that are genetic counselors or anything that is related to that. We will have a day before the Family Forum on April 19 in Nashville that is really meant to connect the clinical care providers together in a nice way and to really start to stimulate an integration of the clinic network. So what are we? And who do we want to be? As you can probably recognize my European influence here, this is the Pont du Gard in France. it is, what do we want to be? We want to be a creative bridge between industry, clinics, academia, and others. And this webinar has come to an end. There is a next webinar that is planned for May 7. The topic is to be announced, and we would like to get your input. Is there anything specific you would like to hear about? And you can email your suggestions to Simon. And now we are ready to take or to answer questions.
I also, as I think most people know, our annual research conference is June 8th and 9th. So while we have on the schedule now a webinar for May 7th, we're hoping to be able to add live video technology to this webcast. And then from our NF conference, I don't know how many people out there are familiar with Reddit, but they have online columns, Ask Me Anything. President Obama even did one, Ask Me Anything. And so what we hope to do at the conference is do a live Ask Me Anything from any doctor you would like to know from NF, any question you would like to ask, and some questions they would answer, some they wouldn't. But that's on the schedule also. So we hope to be able to get the GoToMeeting to get the actual full video working. And that's when we hope to have our one after, the one in June would be live from the NF conference.
Thank you, John. We are now going to turn to the question and answer portion of this evening. We've already had some questions coming in, so we're going to take as many as we can. And if you'd like to ask a question, on the right side of your screen, there's a little chat box. You can send that to you. We probably won't be able to answer everything because we're getting lots of questions, which is a great sign. But we'll answer as many as we can here, and then certainly keep those in mind for future webinars and for the website as well. So,
okay.
First, you want to go?
Yeah,
you want to read the question? Sure.
Yeah.
Okay. So one of the questions that came in is there any new hope for new drug combinations?
The answer is yes, there is. And there is going to be very soon a clinical trial that will be led by Brigitte Wiedemann on MPNST. That is a combination of an HSP90 inhibitor with an mTOR inhibitor. So yes, there are combinations, and that is definitely something we should aim for because of the complexity of the disorder. The only thing is that if you combine, you have the risk also of adding toxicity. So it is a tricky thing to do, but it is definitely something that we're looking at, and we're looking at that also in the NF Preclinical Consortium.
Thank you. Next question. Why are you only, you meaning CTF, why are you only collecting neurofibromas? What about healthy tissue?
We have talked about that, and we have thought about collecting healthy tissue too, but it is considered relatively unethical. And what we do now is we use blood as the normal tissue in order to normalize the tumor variation from one patient to another, but we don't think that we ethically can get that approved by the IRB to use normal tissue from patients.
I would just add, Annette and we use a lot of acronyms. We try to always spell them out. I would start with the CDMRP, Congressionally Directed Medical Research Program, but she just used the term IRB. That is Institutional Review Board, and there are multiple IRBs that you must go through. For example, our NF registry had to have independent IRB review before we could post that online. Any clinical trial must have multiple levels of IRB review, and certainly any tissue collection. So that's what Annette was talking about. IRB approval would probably not get you, unless you could show a compelling interest to collect healthy tissue, it's considered unethical to do so.
Yeah, IRB, and thanks, John, because that's really important. IRB is really a review board that is only there to protect the patients.
Yep.
So they are very worried about everything we do, everything we say, everything we ask. And yes, the registry, the biobank, everything goes through this process in order to make sure that that the patients are perfectly protected.
Next question. How many samples are you aiming to collect?
So for the pilot project, it is a relatively small project, and we are still discussing with the clinician what would be the number. We are thinking about, I think it was 50 patients, 20 tumors, which would bring us to 1,000 samples. But of course, the biobank is an open end. So the idea is that once we have gone through the pilot, this biobank would really become an open source for potential other tumor how you call it?
Here's one.
My son has NF and has a tumor on his optic nerve. Any possible treatments, and how is chemo with these type of tumors?
I'll start with that one. I know Annette can add to it. Right now, to the best of my knowledge, the best of our knowledge, chemo is the only treatment for those. Carboplatin vincristine, which is kind of commonly called the Bakker regimen, has been used to treat those tumors. I mean, I will say about it, and these are low dose and they're commonly accepted in children. So, I mean, while they're decently tolerated as chemo agents, nonetheless, it is not an optimal treatment, or it's the best treatment out there. But certainly, this is an area of keen interest for the foundation. Now, I will say this about optic nerve tumors: is we were recently, Dr. Michael Fisher from CHOP was in here, and he is proposing to us a natural history study. Now, these are long studies, expensive studies, but nonetheless, they're exactly the type of study. I think it was a 5-year proposal on that. Was that what we talked about? Yeah. These are exactly the type of studies that we couldn't have done 5 or 6 years ago, that we can do now, and that we should do now. And while I know your son is of age that you want something here and now, really for children, these are incredibly important things to conduct so that you can address efficacy and toxicity issues. So there are currently approved chemo regimens for optic pathway gliomas, but there is a lot of work being done on let's say the front end and natural history studies to really look at the current treatments, segregate them. There might be genetic differences between different types of optic pathway tumors that respond differently to chemo agents. So that's an area of keen interest as well.
Yes, definitely.
Next question. That is great that TCGA is analyzing MPSNFTs. What about sequencing of neurofibromas? What about whole genome sequencing of NF patients?
So yes, it is already a question that I asked Kenna. The problem is that TCGA today is committed to malignant cancers. And the only malignant cancer that we have in the NF community...
I'm sorry, just for everybody else on there, clear the acronym for them, TCGA.
Okay. The TCGA is the Cancer Genome Atlas Initiative. It is something, it's an institute or a part of the NCI-NIH organization, and they have been sequencing a couple of years ago glioblastomas in order to understand better the genetics of glioblastomas. It has been a huge success. Recently, they have also sequenced and fully analyzed colon cancer, and now the idea would be that they would do a major initiative on malignant peripheral nerve sheath tumors. But the thing is that they are today limited to malignant cancers, so only those are eligible for their analysis.
I would add to that. That is such a great project for them to do for the NF community. And as you get into doing neurofibromas or plexiforms, the cost of whole genome sequencing, the very first full human genome sequence cost $3 billion. That project was co-led or led simultaneously by Dr. Craig Benner and also by Dr. Francis Collins, who discovered the NF gene. So the first one cost $3 billion. Now, Annette and I were meeting with a doctor in Ohio, and I asked him what the current cost is, and he said, well, you can get a 10-pack for $60,000. So it's down to $6,000 per person if you do it in groups of 10. I believe, what we're seeing in genetic testing is a cost curve that is following basically the PC curve. Yeah. I think that will be at $1,200 within 2 to 3 years. And that will open up a lot more ability for groups like the foundation or even individuals to pay for their own sequencing, but somehow it's going to be the collection and analysis of that data that's just going to be vital. Having all that data is one thing, but really being able to do the statistics and tease out what the value of it is going to be the real challenge.
I think one of the other things is statistics is one thing, and I entirely agree with you, John. On the other hand, I think we should look at NF as a machine, a very complex machine, in fact, that we need to figure out how it works. And what we are trying to do as a foundation is really to stimulate cross-disciplinary activities and that people collaborate and look at this machine from different angles. You know, because if you have data, you have huge power. But if you can share the data and put it in the right context, you have the force. And I think that force will drive the cure. So I think we need really multidisciplinary analysis.
And along those lines, a good question that's come in. Approximately how long are we away from getting successful treatments in years?
Yeah.
That's a $1 billion question. I think the question is, how do you define successful? And that is, I think, the major thing. If we will definitely come up with treatments that will do something, but you know, defining successful.
Let me take that from a parent's point of view. And also, I would say there are some treatments out there that, while not silver bullets, have made meaningful changes in improvements in people's quality of life. Certainly Avastin in the NF2 community and schwannomatosis, Gleevec, which has been tried in certain forms of plexiform tumors. So there is progress out there. Now, these have been done in clinical trials. So how do I define success? I define success as an FDA-approved clinical trial. And the ultimate success for this foundation is safe and effective treatments for all the complications of NF. Now, your question was how long for that? And this was a question that I would have ducked a couple years ago. But I will put it out there right now. Here we are in 2013. By 3 years, in 3 years, I am confident that there will be in front of the FDA. I'm not confident it will be FDA approved, but I believe there will be in front of the FDA an application for a treatment for neurofibromatosis. Now, the follow-up question to that, which I'll ask myself, is here's the rub. I don't know which complication it's going to be for. It could be for meningiomas. It could be for optic pathway gliomas. I've seen research heat up in certain areas and get close and then find a closed door and kind of cool off and go back to the drawing board. And I've seen other closed doors open up. But I do feel, and I'm not just giving false hope. For those who know me, I mean, I think part of my job is kind of dampening expectations quite a bit. But I have every confidence that we will have a drug in front of the FDA in the next 3 years. I can't promise which complication it will be for.
How can NF1 funding benefit NF2 and schwannomatosis and vice versa?
Let me start with that. This is a great question that is really, when Annette talked about proof of concept earlier in her slides, there's no better proof of concept in the commonality in research for these two disorders than mTOR. mTOR is an acronym for the mammalian target of rapamycin. It is a drug. mTOR inhibitors are classes of drugs that have been tried in NF1 for years. They show efficacy, and actually going back to the combination question earlier, there's some really recent mouse model work that shows great efficacy when you combine an HSP90 inhibitor with rapamycin. But specific to the question, so now I think it was NF2 is our first funding that tried mTOR on some NF2 tumors that really garnered a second round of funding from the CDMRP. So they all three are common. They are common in that they all are tumor-based disorders. While they all stem from different genetic mutations, there is a commonality research. Certainly, Avastin in schwannomatosis and in NF2 have shown to be efficacious in certain patients. So there's a lot of cross-work being done across different drugs.
Yeah, and I think to add up to that, I think if you do see certain pathways, and if you go to the disease maps, you will see that certain pathways are in common between the two disorders. So compounds that work in one kind of mice model are giving us a certain confidence that they may do something in a whole organism. So they're definitely then of interest to test also in NF2. So I think the cross-contamination, to put it in this way, between NF1, NF2, and schwannomatosis may become more obvious while we understand better how these disorders work and how many pathways are really in common between these disorders. And I would even go one step further. It might be that we see, and that is why the analysis is so important of the pathways that are involved in NF1 and NF2, because the solution may come from a whole different angle, where a disease where we do today do not expect any connection to NF, and suddenly maybe a compound that works in this other disease or disorder may work for NF, and that is exactly what we purpose drugs are for. So I don't think we can cut biology in pieces. We have tried to do that because that is how our brain works, but I think we need to open up our minds and really look at NF as a complex machine with different phases and with different problems, and I'm pretty sure we will find a solution.
Do you have any preferred biological targets for drug discovery?
If you ask me, yes, I do. Of course I have, but I'm not going to say them because I think they may come out of an angle where today I don't expect it. But yes, I'm definitely looking at different biological targets for the moment. We're kind of listing up the different biological targets, and then we try to see what is important enough for neurofibromatosis to be hit by molecules.
Give some examples. What are some of the targets you're looking at?
Well, the problem is that we're talking to companies, and we're in a confidential area.
Got it. Okay.
So that is why I'm a little bit hesitant to talk about it. I hope really soon, though, to be able to tell you something, but for the moment we are under confidentiality with a couple of companies that may have molecules that are really important for NF2 and NF1.
We love that you are a bridge between doctors, patients, clinics, and researchers. Is this bridging only in the States, or is this available worldwide?
I hope it will become worldwide very soon. Today it's mostly in the States, and as John always says, keep focused, Bakker, because I have the tendency to change the world essentially the same day as I started to work here, because I'm really excited to help and to make sure that something happens for neurofibromatosis. But yes, the idea is to connect to the whole world. We had a Chinese PhD here in December at the moment of the gala, and he is interested in connecting us to Asia. We have some good connections with the European NF community. We are funding European NF labs. So I think our bridging is pretty global.
In the research area, we're definitely global.
Yes.
And I would point out that is one of the advantages when we launched our research program back in the 80s. A lot of NIH and certainly the DoD funding is not open to international labs. So it allows us to come in and really see a quality level of research that, they don't have these other options, and they're coming to us. So I think at the NF conference last year, we had 320 scientists and clinicians from 11 different countries. So in research, the collaboration is definitely there.
Yes.
In terms of the foundation working with other international, NF groups, we try. I just met with, it was John Banks came through from Australia last week. We talked to the people in Europe. But really our focus is here. There are only so many hours a day, and so our focus just in terms of, the fundraising activities. Now, I will mention this. One of the reasons why he stopped in was that for those on the phone who know, Cupid's Undie Run, which is a separate organization with very close ties to the Children's Tumor Foundation, grew from 6 cities last year to 17 this year, one of those cities being Sydney, Australia. So that's a great example of a program that we'd like to offer to other European countries, maybe launch that and make that into like a global program. Again, to benefit those areas there, this would not have any immediate direct benefit to Children's Tumor Foundation. But we do work to global, be in touch with the other global NF patient organizations, but I really don't want to overstate the case because it's just, we've got a lot of work to do here. Next.
Are there going to be any research done on the neurological issues that children with NF have, such as executive functioning or autism disorders?
We are currently doing that on multiple areas. In fact, we are working together with Maria Acosta from Children's National on a project that will help parents directly to cope with the neurological problems that their children have, and I hope Maria will be able to announce soon this new program. We are looking at the biomarker approach to look at potential markers for learning disabilities in the blood of children with learning disabilities. So yes, we're definitely looking at multiple approaches to look at learning disabilities for neurofibromatosis.
I would just add, there was an initiative kind of launched here a couple of years ago called LeadNet, which is a learning disabilities project. and we have not been successful. This is a multi-million-dollar project, much like the NF Preclinical Consortium, which we showed you was $8 million over 5 years. That was led by a matching grant by our board member, Dan Gilbert, who basically funded half of that and allowed us to get that off the ground. With LeadNet, we have not been successful yet in attracting a lead donor to get that off the ground. So while we're still doing a lot of learning disabilities work, it's not been as integrated. Right now, our kind of focus is on getting the Sonodos project launched for NF2. So we have this LeadNet proposal here on the shelf. We hope to kind of, get that marketed to some lead donors that would actually make a large gift that would allow us to initiate that, and then we could go out to the broader community.
Any results of ongoing studies, specifically the chemotherapy in treating patients with NF and progressive plexiform neurofibromas?
So for the moment, there are a number of studies that are running, and although there are some hints that some of these compounds may work better than others, the full results are still to come. I think if I'm not mistaken, by summer we should have the results of some of the clinical studies that are ongoing now. And then, of course, the Gleevec study has shown benefit in certain patients of plexiform neurofibroma, but even there, an extra clinical study is ongoing to understand better which are the patients that can really benefit from Gleevec, because that is another study. So the first clinical study is typically a clinical study with mixed population of patients, and then once people see what they call a hint of activity, they enrich the potential responders by patients that they may think will have value from the treatment, and that study will start this study from Wade Clapp, and I think that will be as well.
Yep. Let me just, because we're kind of getting down to the end here, but I want to try to tie a bow on just a couple of threads that have gone through here. Well, as Annette mentioned, Gleevec, and Gleevec has shown some very good potential in NF1-related plexiform tumors, as has Avastin in NF2 and schwannomatosis. But it doesn't work in all cases. In fact, it probably works in less than 50% of the cases. And so I think back to last summer when the Cystic Fibrosis Foundation announced a new treatment called Calideco that actually, and they're a very conservative organization, they do not throw the word cure around lightly, that cures cystic fibrosis. However, it only works in 4% of the patients. They can identify those patients because it works on a specific mutation. So in cystic fibrosis, while there's a variety of mutations that cause it, you all have the same kind of clinical outcome. Your lungs don't work. And in F, it's a little more complicated than that. But this is the intersection, the question about genetic testing earlier, getting your whole genome sequence. Why does a drug work on some people and not another person with the same disease? Likely the answer is in the genetic mutation that is causing that disorder. So that is the intersection. That is why I'm very excited I think that we will have something in front of the FDA, because I think that the promise of the Genome Sequence Project that really completed in 2003, here it is 10 years later, that promise is only starting to be realized today. So I see a time where we are sequencing everybody's genome with NF. I see a time where we know that we can say, okay, you have this mutation, have some Gleevec. You have this mutation, Gleevec will not work for you, try alternate A, alternate B, alternate C. So that's what I see coming down the road.
Yeah, but that will need a multidisciplinary approach.
Yes.
And will need really a hugely collaborative effort across the whole research community in order to look at all these different data sets. Because as John said before, generating data is getting less complicated, less difficult, and less expensive. What we do with the data is going to be the next step. And I think we need million eyeballs to look at the data sets in order to figure out what is it that is really going wrong.
Before we exit, I'll share with you this one final story. So at the conference we're at Faster Cures, the keynote speaker was the head of the X Prize Foundation. And this is a foundation that put up $10 million for the first person to fly into space on a commercial rocket. And when the Deepwater Horizon, the oil rig went down, the BP rig went down the Gulf several years ago, they found a donor to put up a $3 million prize for anyone who could double the efficiency of oil removal from seawater within 2 months. And so the X Prize Foundation was down there, and they were interviewing one of the contestants. And this guy did not win. His contraption only improved the efficiency by 175%. He finished second. I think the winner more than doubled the efficiency. But they're interviewing this guy on the boat. He's dragging this contraption through the sea. And they said, well, how long have you been in oil recovery? And he looked at his watch. He said, are you including today? And it turns out this guy was a tattoo artist in Las Vegas. Now, he was also an inventor on the side. It wasn't just like he took up oil recovery that day. But nonetheless, the moral of the story was open systems, collaborative efforts, big data. You've got to be out there. You've got to open up your systems, which we're trying to do. You've got to be collaborative, and you've got to be open to new ideas. And that's where we're headed. That's where the whole field is headed, and we want to be at the lead of that. So that, that question of when will there be treatments? So I don't have to sit here and say, well, I think we'll have one treatment within 3 years. I'd like to say, everything's done and hand the keys to the next genetic disorder.
Well, that brings us to the end of our webinar. Thank you, John. Thank you, Annette. Thank you, everyone who called in tonight and listened to this. On the left side of the screen there, you have our contact information. Feel free to send us notes. This has been recorded. We'll be putting this up on the website sometime in the next few days. May is NF Awareness Month, as you all know, so be looking to our website, your email box, Facebook. If you don't like us on Facebook yet, please do, and we'll be sharing a lot of information there. And see you on May 7th for the next webinar. Again, thank you and good night.